Hello, everyone, and welcome to another episode of The Tides of Business. My name is Tamara. I am the host. Our incredible guest today is Dr. Jamie Craggs, who is an amazing inspiration. I'm going to try to not fangirl this entire episode, but he is one of my biggest inspirations. He has pioneered ex situ coral reproduction, so I am very excited for him to be part of this episode. So, welcome.
Thank you very much. Thank you for inviting me. I really appreciate it.
Yeah, absolutely. So when we first get in, I kind of want to talk a little bit about like your background. How did you get into coral spawning? Because until I'd heard about you winning Aquarist of the Year with Massna, I didn't actually really know this was a big thing. I didn't really know this was a career. So how did you kind of get into this? What was your inspiration in thinking, heck, I can do that?
Yeah, So I've been working in public aquariums. That's my background for 25 years now. And I've specialized in keeping corals that whole time. So I started back in 98 at London Aquarium, built the first oral tanks in the UK in a public aquarium. As I've always worked with corals in that setting. And I suppose whenever you work with any animal, you want to breed them, right? It shows that you're doing something right and they're reproductively fit and they're ready to create the next generation. So I've always wanted to try and do that with corals, even back in the day of working at London Aquarium. And then, as the technology of the aquarium industry, the hobby industry has sort of advanced so much in the last, 10, 15 years, the likes of having the microprocessors like Apex and so on means that you can have so much control around how you manage your systems. Because working in a public aquarium, we work with many different types of animals. It's not just corals. We breed, seahorses, work with freshwater fish. But I, back in sort of 2009, 2010, I was doing quite a lot of work on breeding jellyfish and working on sexual reproduction of jellyfish. So we were figuring out how do we do in vitro fertilization with jellyfish. And it kind of, a couple of years of doing that, thought, well, they're the cousins of corals. Let's apply how we've been approaching the jellyfish and see if we can do it with corals. So in 2012, we started Project Coral here at the Horniman Museum with the simple goal of could we predictably spawn porers for the first time in the world? While there have been spawning events in people, home aquariums, public aquariums, it had always been accidental. No one had ever gone out and gone, right, what is making a coral tick? And, can we put all those environmental parameters in place to trigger these spawning events, but also importantly, know when that was going to happen. So we could literally be there witnessing the eggs and sperm coming out of this first acro spawning. So that we put the project in place in 2012, and we had 8 months later our first predictable spawning. And it really, that was pretty foundational, achievement, really. And it really solidified in my mind that we were absolutely on the right track. It created the platform that has just exploded now. And we have, I think we've now hitting about 52 species that we've managed to spawn, which is pretty amazing, from Caribbean to Indo-Pacific to, Red Sea. So, once you've got the platform and you've got the predictable spawning, it just opens up a whole world of research opportunities, thinking about climate change. How do we increase productivity from these spawning events to rebuild reef? How do we create sustainable aquaculture markets for the aquarium trade? All of these are now possible through the reproductive work. So, it's been a journey of discovery. We're still learning all the time. It's fascinating. Just when you think you've got it all sussed, there's stuff comes out of nowhere and you learn something new and it takes you in a new direction. So it's pretty cool stuff.
Yeah. Oh, that's amazing. So you co-founded the Coral Spawning Lab. So what was the concept behind that?
So I started Project Coral in 2012. We'd had our first couple of spawns, a couple of years of spawning. I then started my PhD 4 years later at Derby University with Professor Mike Sweet. I'd been supporting him during his PhD on coral disease, and then he got his position at Derby University and said, why don't you write this up as your PhD? And so I worked with him on the PhD, completed that in 2019, And we were just getting so many requests from different research groups all around the world asking for advice about how to develop their research potential, how, their restoration. And so we just thought, well, let's co-found the Coral Spawning Lab. And so 2019, that started. And we built systems in the UK and shipped those out to various places around the world to support whether it's universities or NGOs. And we genuinely thought only one or two systems might go out. We've done no advertising. In fact, we put one email out on a list server, and that's the only bit of advertising we've done. And it's been absolutely crazy. 4 years, over 70 systems have gone to, I think now 15 countries around the world. So it's been exhausting, but it's amazing seeing the capabilities and the capacities it's allowed other researchers to do. So, and I would say probably 50% of those are using it for pure research. So just understanding coral reproduction at a better level. A number of colleagues are working with something called CRISPR-Cas9, which is gene editing work. Not thinking about genetically modifying corals to go into the ocean, but really trying to fine tune which genes are responsible for increased thermal resilience, so which corals might be able to withstand future climate conditions. And we need to be doing these really fundamental genetic analysis to understand where that potential lies. The challenge is if you have to do it, the CRISPR-Cas9, you have to inject the egg, so you need access to the spawn. And if you're only getting spawning once or twice a year, it just really slows the speed of development that you can develop these genetic tools and these understandings. So, one of the first big projects that we did for Kaust University in Saudi Arabia is we designed a system that could give them, on theory, on paper, it would give them 24 spawns a year. So it just would accelerate their ability to do the CRISPR-Cas9. It actually went way further than that. The system we designed in the first year, I think they got 96 spawning events. And so it just absolutely exploded the potential of accelerating these really cutting edge genetic tools. So yeah, I mean, and like I say, like probably half of these systems that got out there to support amazing research like that. The other half is looking at how do we upscale restoration. We need to be delivering restoration at bigger capacity, bigger scale that make an ecological difference. So, and these are some of the big challenges that lots of groups around the world are working on, really.
Yeah.
Yeah.
And you've been a part of a lot of really big groups as collaboration. Are there any specific knowledge sharing groups or collaborations that you've been a part of?
Yeah. So I sit on a board with the Coral Restoration Consortium, looking at land-based coral spawning and its capabilities. So while I don't work with any Caribbean species over here, it's sort of working with the likes of Kerry O'Neill and all the team there that are physically using these tools to restore the Florida Reef Tract. I can provide my input on, I've used a latest study to look at feeding and how that's had an impact on post-settlement survival, and can we then apply that to their work. So, the work we do behind the scenes, I mean, there's a tiny corridor up here, but we've squeezed in systems. The way we always see it here is we're trying to develop new knowledge that we then offer out. So we try and publish everything open access. So, you can go and download, anyone on the planet can go and download that paper and read the sort of toolbox. And many people have, like the original the original spawning system. I know different groups have just taken that paper and modified it and used it for their own ends. The Coral Spawning Lab was then offering something that was off the shelf that they could come and buy it from us, and rather than and with that, they don't just buy a system, they buy us and our knowledge. So we all work with the teams to kind of train them, and so there's a little bit more interaction from that. So yeah, I mean that group in the Caribbean has been a big one. We've worked with groups in the Maldives. I've worked with a group in Qatar University. They're developing their restoration potential. We've got a lot of projects coming along the line in next year, looking at sort of expanding some of the work in the Indian Ocean. So there's a number of different things from the ornament side of things. We do research behind the scenes. We're working with some groups on a project in the Indian Ocean, and then the Coral Spawning Lab is then working with different groups as well. So there's a number of different routes of ways we're working, really.
Yeah, that's simply amazing. I'm really impressed with like how many different groups you're able to work with and kind of, share your knowledge to kind of help the entire coral restoration grow throughout the world. It's pretty amazing.
Well, I mean, at the museum here, so I'm the principal aquarium curator and I oversee the collection. Because of trying to work with more groups, I've been winding down my hours here. So over the last year, I've reduced my working week from 5 days to 4 days to give me a bit more capacity. I'm doing that even more. So I'm reducing my hours down to 2 days a week. And then I'm starting with Mars Sustainable Solutions. So Mars Incorporation owns 200 different companies. They are, not many people know this, but they are funding one of the biggest restoration programs on the planet. They've already planted 1.3 million corals on reefs from Indonesia, Maldives, Hawaii, all over. So I'm joining the team there. So it will give me an opportunity to get into the field an awful lot more, which will be great. Because I mean, the work we're doing behind the scenes of the museum is fantastic, and it will continue. We're going to develop that and build it, but without sort of planting the coral back in the ocean, it sort of doesn't allow us to complete that full circle. So working with Mars allows us to, to look at some of that, really.
Yeah.
Yeah, it's
a lot larger scale for sure.
Exactly. Yeah. So it's going to be really exciting. I can't wait.
That's pretty amazing. I was actually going to ask you, like, what percentage of the time you spend in the lab versus how much you spend, on site? Because are you still in Qatar right now?
No. I'm in London at the Ornament Museum. So this is our tiny lab where we do all the water chemistry. We look after the corals. So when spawning happens, we've got a spawning event coming up in about 3 weeks. This sort of black tarp here gets rolled down, and we're going to heat up this room. It's starting to get cold in the UK, so we'll heat up the room to a nice tropical temperature. And all along these benches, this is where all the fertilization work is going to take place, and for the various experiments we've got planned for this year. And then literally just up the stairs here are 4 spawning systems that this year we're replicating. Actually, there's 5 spawning systems, but 4 of those are based on tropical regions. So we've got a Fijian, an Indonesian, an Australian, and an Indian Ocean system. And then we also have, we started doing some work with cold water corals. So we're working with corals off the UK coast, or Gorgonian, and that's our 5th spawning system. So, we managed to crack some British species spawning this year. We've got a really nice experiment with a PhD student in the UK looking at larval settlement and something called competency period. So how long can the larvae stay in the water column for? So there's going to be some really nice work coming out of this over the next couple of years.
Oh, that's amazing. And just kind of like a high level question, I guess. So, obviously we know the oceans are warming, the corals aren't handling it as well. How do you know what corals to kind of look for specifically? Are you looking for corals that kind of do better at that warmer temperature? Is that kind of where your focus of research on? Is it, or just whatever colonies you happen to have to work with? Like, how do you select what corals?
So we do a little bit of thermal resilient work at the museum, but we've also partnered. So another one of my PhD supervisors, Dr. James Guest at Newcastle University, he and the program of this is just finished, but it's been 6 years European funded project called Coral Assist, and all of this work was done in Palau. And I got invited out to go and join the team for a couple of years in a row, where we were collecting corals, and then Dr. Adriana Humanas is one of the postdocs on it. She's amazing. She was leading all the thermal resilience side of things. So for an acroporid species that we were working with there, Acropora digitifera, she had marked 100 colonies, taken fragments from those, and then done very controlled thermal stress on them all to rank them. So you could rank which ones had more resilience, which ones had less resilience. And then during the spawning event in 2018, we basically were, through that initial experimentation work, we knew which ones were more resilient, which ones were lower resilient. And so we took eggs from the high resilient, sperm from the low resilient, made that cross. We took eggs and sperm from high resilient, made that cross. We took eggs from low resilient, sperm from high resilient, and then low. So made all these pairwise crosses, settled those. Adriana then did stress experiments on the juveniles to see whether they inherited those, so the F1s. And then in 2020 and 2021, those individuals were sexually mature and they spawned. Actually, 21 and 22, sorry. Those individuals were then sexually mature, they spawned, and she's carried on that work to look at the F2 generation. So they've literally just published this work only 3 weeks ago, and it shows multi-generational inheritance. So it's the first time that's been shown that actually if you selectively breed, it doesn't just stop with the F1, it can be passed down. So it creates a really, really important, tantalizing glimpse about the potential that we can do with this. James is incredibly modest and also, and rightly so, very cautious to say that the thermal gain that was bred was modest. It's not going to solve the world's problems. It's the first step in understanding that, but it did show an increase. So, and some of the work we've done here is looking at then we've just got a paper that we'll be submitting soon, looking at what you feed a coral will also determine the level of thermal resilience that it can gain through feeding, not just through the genetic route, but also from what's called the epigenetic, the environmental routes as well. So there's a lot going on within these dynamics, I suppose.
Really, I wouldn't have equated food to helping with like the thermal resistance. So is it at all stages of life, or is it just like at that very young larval stage?
No, so what we did for this experiment, we started this in 2019. Rather than doing those pure pairwise crosses, we just mixed multiple genotypes together because we weren't selecting for the genotype, we were looking at the environmental conditions. So we created a complete mix of offspring that were either completely related, half related, or completely unrelated, so that we could then see the environmental impact rather than genetic impact. Settled those, then grew them for 18 months before the thermal stress. Those were fed on a specific diet from the day they settled for 18 months every single day. We isolated the systems for 2 hours a day to feed them specific feeding regimes. And then on that 18-month time period, then subjected them to, because ultimately it's a death experiment. You run it right the way through to see how far you can push them, and then record, the more or the certain types of food, how much further can you push them on their thermal resilience. So yeah, you can't do it at like multiple time points because you end up running out of corals to do any work on, basically. So yeah, it was a fascinating piece of work.
Yeah. So I was reading that you guys also breed urchins, specifically tuxedo urchins, to kind of like clean up the algae when your baby corals are growing. So you're growing your baby urchins with your baby corals. So I spoke to Martin Moe about it. That was always his dream, was to have baby urchins with baby corals. Did that idea come from him, or did you kind of create that like on your own?
So one of my chapters, my PhD, I always wanted to look at the influence of herbivory. Originally, I was thinking about using hermit crabs, but then, one of the issues is crustose coralline algae, CCA, ultimately grows over the juveniles, and we know urchins are brilliant raising everything back down to rock, including CCA. Martin provided me with so much support and information in terms of how to go about doing this. So yes, he was very instrumental in supporting this work. We initially were going to look at diadema because of Martin's work with diadema antillarum, but we can't get hold of that in the UK. So I actually ended up ordering the smallest diadema setosum that I could get. So they were probably about the size of my little fingernail here. And my initial trial, they just basically moted all over the corals and ate them all when they were juvenile. So we had to, that did not work, no. So we had to go even smaller and have even smaller urchins. So, and we've continued doing this. But yeah, about 2 months before spawning, coral spawning, we will spawn the sea urchins because that gives enough time to get the urchins through their pelagic phase. They can then settle, grow out, and at that point they're about 1 to 2 millimeters, which is the perfect size to then introduce with the juvenile urchins, so juvenile corals. And it worked wonderfully. That experiment ran over a 6-month period. Basically, the more urchins we put in, the higher the survivorship of the corals. So we had an 8-fold increase in survival. And we've carried on doing a lot of work with urchins. Yeah. I mean, it's really important, right? You get more corals. If you've got more corals, you've got more material for restoration. The lines I put in the discussion of that paper was, so we were doing it with tuxedo urchins, which, it's a good urchin for the aquarium trade. So could you, if you were to do this in a tropical location where you're trying to create lots of coral for restoration, could you grow the urchins alongside them to get more coral? But then once the urchins got big enough, could you sell those into the aquarium trade and bring an income source into restoration? We started thinking this a bit further. Well, what if we took, in the UK, we call them the collector sea urchins, Trypneusses. It's the orange spine one. I think they call them Halloween urchins as well. Yeah. But they're used for the human food market, right? So we thought, well, what if you could spawn Trypneusses? Again, for restoration, you create more corals, but then grow them out and sell them into the human food market, again, bring in income source. And so last year, I got a small grant, and my deputy, Hartley, earlier this year, went out to Palau, again through James Guest, and working with the Palau Coral Institute, Palau Coral Reef Institute. We used the grant to build a small phytoplankton and urchin culturing facility in Palau, where we're literally actively trialing these right now to see if we can generate that circular economic model, really. So how do you bring income that funds the restoration, which means that it has longevity rather than just relying on constant grant applications. So, and the team out in Palau are doing a great job with this new lab. They're culturing phytoplankton. They've just had we literally got a message yesterday. They've got their first settled urchins. So, it's going to be really cool work coming through over the next couple of years on that front as well.
Yeah, that's amazing. I'm glad you brought up like the funding aspect, because I would imagine that is probably a pretty big bottleneck with the research, is how much funding that you can get, not just in a lab, but like worldwide for research. So is it mostly just grants, or, do you have people that donate to further the research?
Yeah, I mean, we sort of sit somewhere in a weird place in the fact that we're a public aquarium. We don't do restoration ourselves, but certainly thinking about the restoration community, I think there's loads of people, different models that are trying to approach it in different ways. You've got completely grant-giving organizations or grant-funded organizations, and that can be very precarious. You've got others that are engaged in the tourism industry. You've got others that have gone fully commercial and pay, they offer their services for a fee to a resort or something like that. So I think the challenge with the grant funding model is the fact that grants are only good while the topic is in favor at that time. And does that have enough longevity to make sure that it's sustainable? Particularly when you're a lot of the restoration work that's happening around the world is using asexual fragmentation, and it builds 3-dimensional structure very quickly or comparatively compared to sexual reproduction. But so that's a positive. It's really good, and it should always have a place in the restoration toolbox. The sexual reproduction brings that genetic diversity, but you're obviously starting with something that's a millimeter across, and it just takes a long time to get to the same structural complexity that you can do asexually. So, and if you've got a grant that only lasts 3 years, it's very difficult to prove how successful you've been in a 3-year program. And that's perhaps some of the challenges that are faced out there.
Yeah, I would definitely think that's the biggest thing with the research. It takes a long time to set up the systems, get the corals ready, to get the spawning, and actually make some sort of meaningful impact on the research in short grant, time cycles.
exactly. Yeah.
Yeah. So I always have people, and I'm sure you get probably inundated with emails and people asking, but how do people get involved in like coral reproduction as a career? Like if somebody was like, oh, I love this, I'm passionate about this, I'm very excited, like how would somebody go about that?
I would say there's probably a number of different routes to starting with the home system. I really try and advocate for many people to start playing around with this in their home system. I put a spawning system in my kitchen after my PhD, just for a bit of fun. I could do stuff at home that I can't necessarily do at work. And we spawn 15 species at home now, and 3 of those are new to science. And it's honestly, it's not a big system. It's 700 liters. It's got a tank this sort of size at the side of it that I do all the embryo rearing. So I think, that's a starting point that everyone, and I think this is where the hobby will go over time. I think more and more people are wanting to pick this up and play around with it. I think the potential of thinking about all of the different color variations that you can produce on the back of this will be significant in the trade as we move forward. I have no doubt about that. So that's one angle. That's, either from a hobby point of view or then going into an aquaculture for the aquarium industry point of view. That's one route. Obviously going through university and looking at studying tropical marine biology is another route. Specifically thinking around, undergrad, master's programs, PhDs. There's a lot now available for the focuses on coral reproduction, and amazing institutions all around the world that do that. You've got Australia Institute for Marine Science, you've got Cal State University, you've got the big universities throughout America working on this, from Miami to Carnegie. There's a lot of opportunity from pure research side of things. And then there's the resort side as well. There's increasingly more resorts are engaging with restoration. There's a lot of opportunity in the Maldives, for instance. Loads of programs, not even just the Maldives. You think all through the Caribbean, from Reef Renewal, have got multiple sites throughout the Caribbean focusing on restoration. So I think there's multiple routes, whether it's pure research, whether it's a hobby land-based, or whether it's applied ocean-based work. There's 3 strands there, all engaging with it at different levels. Yeah.
Pretty amazing. Now, you said something that kind of struck me a little bit. So you said you kind of see this coral, like sexual reproduction, being an impact in the aquarium hobby. Like, do you see people being able to purchase like reproduced corals that have not been from the wild? Like that's kind of the plan that you think?
Yeah, absolutely. It's happening. I've worked with colleagues out in Australia, that Monsoon Aquatics and Interfish Platy. Lou out there, who was at Interfish, he's now over at Monsoon. He's been doing some amazing work, and those are for sale in America now, that have come through a sexual. Yeah. So he's done multiple species. So absolutely, my concern with this, and there's nothing you can do to stop it, it's going to happen, is I would always advocate it should be done at source country, because there will no doubt be coral spawning farms developed for aquaculture in Europe, in North America, and I think it's a crying shame if it cut out the potential sustainable livelihood development in source country. So I would always try and advocate that that's where it should be done. And, if it's done right, it can be done very well. And it just creates a healthy, thriving industry for the future where everyone is benefiting through that chain. And I think that's an important approach personally. And I'm always mindful of, feel very, privileged and proud of where this pool of controlled spawning has gone. But I am very mindful every time I try and publish something that is open access, is opening more and more doors that will ultimately potentially cut out those source countries. And there's nothing I can do about it, but I would always try and advocate that, doing that in source country is the way forward. In fact, Mike and I applied for a CORDAP grant. So CORDAP's a big funding body that was set up a couple of years ago that was or is funding sort of blue sky work around restoration and reefs in total. And we secured a grant working with the Indonesian government where the grant paid for the systems to go out. So they landed in Indonesia about 3 weeks ago. And we'll be working with the Indonesian government to train them in LPS spawning practices so that ultimately they can train up multiple farms in the region. So it'll be working with, acanthophilias, trachyphilias, all of the stuff that, desperately needs that sustainability angle brought into it, but very much working in source country. So that grant it's got another 3 years to run, but within that, they will have the toolbox of developing that up. Yeah.
Oh, that's amazing. So you're talking about LPS corals. Is there a major difference between SPS and LPS as far as settlement?
Not really. They, we're learning something new about every spawning that we sort of do. I mean, back in my kitchen system, I spawned the Homophilia australis a couple of years ago, Catalophilias, Trachyphilias now. The bundles that are produced are massive by comparison to something like an Acapora, because it's just obviously got a single polyp or a few mouths rather than coming out of hundreds of mouths on a branch. So the bundles are huge, but it's a very general rule of thumb. But ultimately, the size of the egg broadly dictates the speed of development. So if it's 400 microns, it will be roughly 4 days of development for the larvae for when it settles. Most of the LPS I've worked with are much quicker because they're quite a small egg. So you get settlement or their larvae within 2 to 2.5, 3 days. And so they're dropping out of the water much quicker than Acapora, and they're much smaller. But there's definitely differences in settlement orientation. So some species actually like settling on the underside, some will go on the top, some will be on vertical surfaces. So there are subtle differences, but the approach is broadly the same. They have to acquire their symbionts out of the water once they've settled. That they are subjected to the same problems that Acaporas are subjected to. That post-settlement survivorship, you get a lot settling, and then a lot dying ultimately because of algae and ciliates and bacterial infections, all those sort of things. You have to manage all of that in exactly the same way, really. Yeah.
That's really interesting because I always hear so much about SPS, but I don't hear very much about LPS. I saw your post on Facebook, I think it was yesterday, about the scalimia that you had spawned, I think a couple of years ago. Besides that, I don't really see very much.
No, and that's why I wanted to do it. I mean, ultimately, you've got a species that, one of my Magna talks I gave was all about LPS spawning and what I've learned within that, because I think, it was always thought that something like a Homophilia, how old is that? Is that, 20 years old and therefore really, really vulnerable to overcollection? Until we do those fundamentals about growth rates and stuff like that, you really can't answer any of those questions. So that was the initial goal. One, nobody had understood when they spawned. So it was an expensive personal sort of hobby to try and do, because I basically had to sacrifice them as they come in. That's the general way that you figure out when a species spawns, is you break them in half and look at the stage of egg development. If you did that every single month through the year, then you would define when that species spawns. With the scollies, they're just expensive. So when they arrive and you divide it in half, you're literally just burning money. But it created the foundation of knowing when they spawn. I then, it's a paper I really need to get around to finishing and writing, but I've documented the full embryological stages of both Homophilia and Catalophilia, and I've done that on scanning electron microscopy. I've done early settlement experiments with that, feeding experiments, and then recording growth rates over time. So when I finally get around to writing it, it will create the blueprint to say, well, this is the speed of growth, this is how you go and spawn them. I just need a bit of time.
You just need a little more time. You got nothing.
I know.
That's amazing.
Yeah.
All right. Well, I want to thank you so much for your time. I really appreciate you, talking about coral reproduction and all the work that you're doing. It's absolutely inspiring to me. Is there any last tidbit or word that you would want any of our listeners to know?
Um, I think, ultimately reefs are, they're facing some pretty tough challenges ahead, and we need to get the message out there about how beautiful these ecosystems are. We need to support the conservation of them so that we can enjoy them for future generations. That's would be my take home message.
Yeah, that's pretty.
Yep.
All right. Well, thank you so much for your time. I really appreciate it. You are all inspiring, and I cannot wait to continue following all of your amazing work around the world.
Thank you very much for inviting me. I really appreciate it.
All right. All right, everyone. Well, thank you very much for joining us for another episode. Stay tuned for next week where we have another incredible guest.